In this video, we're going to look at our second topics for photosynthesis, photosynthesis part 2. We're still looking at topics 3.8 and 8.2 for IB curriculum. We're going to start out today by looking at light absorption in photosynthesis. Now, sunlight produces white light. It produces white light, and that's a mix of all different sorts of colors. And we can actually separate that out in terms of visible light that humans can see. We can separate that out into different colors. And those different colors are derived or caused by different wavelengths of the light. And so reddish orange has wavelengths around 700. Violet and blue have wavelengths of around 400. And the distance of the wavelength is actually proportional to the amount of energy. The shorter the wavelength, the increased amount of energy. So gamma rays, which we cannot see, have a lot of energy. Radio waves, which we're not able to see either, have not as much energy in comparison. And so if we're looking at visible light specifically, light that we can see, the purple-blue light has a lot more energy than the red-orange light, which kind of comes into play a little bit more when we start looking at how light is absorbed by plants in the chloroplast. So when we're talking about light, why is it that we see plants, why do we see them as green? Plants are primarily green. Why do we see that color? And the reason for that, the explanation for that, is plants, specifically the chloroplasts, are absorbing white light. So they're absorbing all different lights, and they use some of that light during photosynthesis in order to carry out the process. Light that's not used is reflected back out from the plant. So it's removed from the plant, or it's reflected from the plant. And that light that is reflected is actually green light. And so that's why we see plants, chloroplasts, as green, is because that light is being reflected or refracted. That's not being absorbed by the plants. Within chloroplasts and within plants, chlorophyll is the primary pigment used in chloroplasts that actually absorbs light. And we can actually create graphs for this, and we can see what colors are being absorbed. The primary colors that are being absorbed are the violet blue and the red and the oranges, a little bit of the lighter blues. But green is really not absorbed at all, and so that's, again, why we're seeing plants as a green color, is that green light is being reflected back. Here's another graph that we can look at. This is the absorption spectrum graph of different photosynthetic pigments, and there's a couple different types of pigments. Chlorophyll A has a different range or different wavelengths that it absorbs in comparison to chlorophyll B. And so chlorophyll A and B are the primary pigments or absorption tools of light. They're most effective in the violet, blue, and the red light. So we've got this region here and this region here that are the most effective, that actually absorb the most amount of light. They differ slightly in what they do absorb. Chlorophyll A is a little bit more of the violet. Chlorophyll B is a little bit more of the lighter blue. Same thing in the orange and the red. And there are also some accessory pigment absorptions that pick up a little bit of the violet blue and a little bit of the blue-green. But you can see right here in the primary green color, there's really no absorption. And again, that green light is being reflected. The overall rate of photosynthesis can be measured in a couple of different ways, and that's by either the production of oxygen, the uptake of CO2, or an increase in plant biomass. And if we are measuring the amount of oxygen that's produced during photosynthesis, again, one of the inputs of photosynthesis are sunlight, H2O, and carbon dioxide. And so one of the outputs, one of the products that we get is O2. And we can actually measure that using some different probe-type software and hardware. We can actually measure how much oxygen is produced during the process of photosynthesis. Another way that we can measure it is also pretty much the same in the terms of the production of oxygen. We can also measure it by the uptake of carbon dioxide. So during photosynthesis, CO2 is removed from the environment, and that's used in order to create the sugars. The removal of that from a system can actually be measured by a couple of different ways. One would be increases in pH, which can be measured in water. And then we can also do the same as we did with oxygen. We can measure CO2 values by using probe, like veneer probe type software and instruments to make digital measurements of that. The third way is by an increase in plant biomass. If plants are harvested at a continual rate, we can actually calculate the overall biomass. Obviously, this would be a little bit more complex, a little bit more complicated than just measuring the amount of oxygen being produced or the amount of CO2 being removed from systems. The use of biomass provides an indirect measurement of the rate of photosynthesis. Now, the second portion of our video today, we're going to look at, get into the steps of photosynthesis. And in this one, we're going to look at the light-dependent reactions. And this means that it's the portion of photosynthesis that requires light in order to occur. The light-independent reactions on this portion are actually the production of the sugars. The end product, the primary end product that we're producing here. But the light-dependent reactions are just as important as ATP and NADPH are produced that allow the light-independent reactions to occur. So we're going to look specifically in this video at the light-dependent reactions, and in the next video we'll take a closer look at the light-independent reactions. So in this image, it does a really good job of kind of summarizing what's happening during the light-dependent reactions. This is occurring within the thylakoid, and the chloroplast is absorbing light. And that light absorption strikes specific pigments within the thylakoid that are basically boosting electrons to a higher energy level. So here's our light, and it's absorbed and bounces across a number of different pigments here. And what that's doing is it's exciting these electrons, and it's going to move them to the primary electron acceptor. Now, during this process, H2O is required and needed, and that energy is going to split our H2O molecule. And so what we get from this is 2 electrons that get sent to the primary electron acceptor that will be used in just a minute. We get 2 hydrogen ions that are going to be used in our electron transport chains, very similar to what we saw in cellular respiration. And we get oxygen. When this occurs twice, we'll get 2 oxygens, and so O2 is created and released from the chloroplast and from the cell, and also from the plant, and that's what we end up breathing in. Now, once these electrons are excited and they're at the primary electron acceptor, they move down an electron transport chain. And this is very similar to what we saw with cellular respiration. Basically the same idea. These electrons are moving from one enzyme to another within the membrane. And during this process, in the same fashion, using chemiosmosis, ADP and a phosphate are bonded to produce ATP. So there's a high concentration of hydrogen ions that collect. As they move across that membrane, ATP is produced. And that ATP is then going to go on and be used in the Calvin cycle. So we're going to use that later. The electron transport chain, the electrons that pass through that are deposited in photosystem 1. Photosystem 2 is the first one. I'm not sure exactly why it's named photosystem 2 and photosystem 1. That's the name that they've been given. And so in photosystem 1, our second one, these electrons are deposited into a second set of pigments. This happens to be the P70 pigments. And here in our photosystem 2 is the P680 pigments, measuring light, absorbing light, at different wavelengths. And so the electrons that are deposited here again move through these pigments, and then with more light, so more light is absorbed, they're excited to a higher level of energy, and those electrons are going to be moved through an electron transport chain again. So there's a second electron transport chain. And during this process, the enzyme NAD+ reductase reduces NAD+ to form NADPH. That is a molecule that's very similar to NADH, and is a highly reactive molecule that's going to be used in the Calvin cycle. And so the whole process of these light-dependent reactions is the production of ATP and NADPH that are going to be used in the Calvin cycle in order to drive that process. That is the purpose of the light-dependent reactions, and they're dependent on light being present. So the light is providing the energy for these electrons to be moved to higher energy levels and then move down the electron transport chain. I have a list of these that we'll walk through, these steps in a list format. So the light strikes pigment molecules in photosystem 2, and it boosts the electrons to a higher energy level. This process continues, so more light is absorbed until electrons reach the P680 pigment molecules. So there's a number of different pigments that they have to go through to start. and eventually they reach the P680 molecule, and they're moved to the primary electron acceptor up at the top. An enzyme catalyzes the splitting of water, and this is photolysis, to release hydrogen ions into the thylakoid lumen. Oxygen is released out of the cell, and the electrons are sent to the electron transport chain. Continuing with this, the electrons move through the electron transport chain. It's similar to what occurs in the mitochondria, and as they move or fall down the electron transport chain, this provides the energy necessary to form ATP from ADP and phosphate. As more light is absorbed in photosystem 1, it activates the photosystem 1 primary acceptor, where those electrons are going to be accepted. And those are accepted from the electron transport chain from photosystem 2. So the electrons that move down the electron transport chain from photosystem 2 get passed to photosystem 1. They're passed through the second electron transport chain, and then we get the enzyme NAD+. Reductase reduces NAD+ to NADPH, and it removes the hydrogen ions from the stroma. So to kind of summarize all of this and what we're seeing happening, light energy is used to excite electrons. The photolysis of H2O provides or produces hydrogen ions, electrons to be used in the electron transport chain, and it releases O2, oxygen, from the cell and the plant. Produced is ATP and ADPH, which are going to be used in the light independent reactions, the Calvin cycle, and this is the production of the actual sugars, and we'll take a closer look at that in the next video. To finish up this one, I'd like to take a look at both the structure and function of a chloroplast. We've talked about the structure of the chloroplast and looked at some of the parts a little bit. Here's another nice clean image that gives us a look at the different parts specifically. Again, the thylakoids, these individual stacks, kind of like the quarters, a stack of them makes something called a granulum, so it's a large stack of the thylakoids. The stroma is kind of like the cytoplasm of the cell, the empty jelly-filled space, and this is where the Calvin cycle is going to occur. We have an inner and outer membrane, and then the lumen is the area inside of the thylakoid. The thylakoid has a large surface area, so kind of like the cristae and the mitochondria that we talked about during respiration, a large surface area is necessary in order for light absorption. This is where the light is being absorbed, that the plant cells, the chloroplast specifically, absorb, and so an increased surface area is going to provide more area for light to be absorbed. The small space within the thylakoid allows for a high concentration gradient of protons to be produced that are going to be necessary in that electron transport chain. And lastly, the stroma, the jelly space outside of the thylakoid, allows for the enzymes necessary for the Calvin cycle to react and to carry out those processes that we'll see in our next video. So in the next video, we'll finish up our discussion of photosynthesis. We'll look at the light-independent reactions as well as the absorption spectrum a little bit, more specifically how light is absorbed and how it's used and how we can measure that.